Hello, I'm Dominic Otto, a postdoc at the SETI Lab and the Fred Hutch Cancer Research Center, and I want to present you our work to develop a new method that measures active and silenced regions in single cells and the computational aspect of it. The method is called single cell cut-and-tag 2-for-1, and we want to target two specific epigenetic marks here. One is an active mark. That's where polymerase II is bound. The genome is accessible or maybe even actively transcribed. And the other mark is H3K27ME3. That's a histone modification that stands for a silenced region of the genome. The method works by using antibodies that in turn attract other antibodies that are tethered to transposases that cut the genome in the vicinity of our marks of interest. And as a result, we get fragments that can be sequenced and then realigned to the genome. And the interesting aspect here is that the fragment length can vary depending on the thing that we are targeting. So here I show a fragment length distribution of cut-and-tag experiments using only one of the two targets. So for polymerase, you can see that the fragments tend to be much shorter, since they are not protected only by the transcription factor or polymerase that's bound here. And fragments from another single antibody experiment, ones that only target the histone modification here. And because the genome is protected by the nucleosome it's bound to in the vicinity of the histone modification, it is protected from cuts, and the fragments that result from it tend to be much longer, but not all of them. And when we put both antibodies on target both marks at the same time, we seem to get a combination of fragment length of those two assays. And our hopes is, of course, to use the distinct characteristic fragment length distribution to recover which fragment came from which target. However, those fragment length distributions are also subject to variability from experiment to experiment, so our approach must account for that. This is some example data of one of those experiments where I just show a small fraction of the genome where a lot of reads or fragments have been aligned to. This is the read coverage, a very familiar way for people to look at this kind of data. And it basically, the signal indicates how many fragments are overlapping at any given locus. The only thing I did here as a preliminary deconvolution is I split up the track into long fragments that are longer than 120 base pairs and shorter ones, just to show that there's already a slight difference in those two signals. But since the fragment length distributions induced by the two targets tend to overlap, it's not a very clear deconvolution yet. The main point I want to make here, though, is that the fragment coverage is hard to interpret, because if two cuts in the genome are, whether they are connected through a fragment or not, it doesn't really make a difference. The only thing that's interesting is how close they are, because the closer two fragments are, the more likely they've been induced by the same target, no matter if they're connected through a fragment or not. So another way that we like to look at this data is through the density of cuts, or the quantity of cuts within a given vicinity. So down here, the signal height represents the number of cuts in the genome per 100 base pairs. And we just use a coronal density estimator to do this. And additionally, I split up the signal into colored bands, where each color stands for a different fragment length that is associated to those cuts. And here you can already see that there are some peaks that are mostly composed out of short cuts, and other areas of high signal where a lot of long fragments are involved. So there's definitely something here, and we want to find a clever way in order to decouple those two signals again. So when we look at the data in terms of cuts on the genome, we can basically talk about a two-dimensional probability density for cuts. One dimension being the location at which the cut occurred, and the other dimension being the length of the fragment where the cut came from. Now, a key assumption here is that the resulting distribution is a combination of cuts that have been induced by the histone modification and another one that were induced by the polymerase. And there's a major difference in the marginal length distribution of those cuts, and also a slight difference in how pointy those cuts are. Because we know that polymerase-specific cuts tend to aggregate in very small peaks, where in contrast those histone modification-induced cuts are more spread out and mark rather large silenced domains of the genome. Now we would like to use this model to do a Bayesian inference and reconstruct the target-specific cut density functions. And to do this, we assume that this two-dimensional cut probability density is a product of two statistically independent marginal distributions that we can in turn model completely independently. Let us first look at the fragment length marginal. So this is how the data looks like for different assays. And we don't care so much about the wiggles, so we just want to model fragment length distributions that look like this. So in all experiments, no matter what the targets are, we always see a combination of these modes in the fragment length distribution, which is of course associated to the lengths of fragments that are protected through nucleosomes. These being the very short ones, being the unprotected ones, and a little bit longer when the fragment wraps around nucleosome once. And then there are even modes for larger fragment length when it wraps around multiple nucleosomes at a time, and is therefore protected and can only be cut in between the nucleosomes, basically. So in order to model this, we thought, why not use a linear combination of some log normal distributions? I mean, they're strictly positive, and we can fine-tune them to have a similar shape to what we see in the data. And then the flexibility for the fragment length distribution only comes from the weights that can be sampled according to a Dirichlet distribution. And with those weights, we also have the option to strongly inform how the fragment length distribution can look like through Bayesian priors that would favor certain modes. Let's say for polymerase-specific reads, we would favor shorter reads. However, it turns out we don't really need an informative prior here. We will later be able to actually find a posterior distribution for those fragment lengths just based on the co-occurrence of certain fragment lengths throughout the genome. And this already constitutes one of the two marginals. And the other marginal, this is where the Gaussian process comes in. And this is really helpful here in the way that we can basically use the neighborhood of any given cut in order to inform its target. So if all the neighbors are already known or very likely to be from one specific target, then the cut that's very close to this neighborhood is more likely also from the same neighborhood. And this proximity information can be very well used through a Gaussian process. So plugging everything together, for the fragment length marginal, we have a linear combination of log normal distributions with weights sampled from a Dirichlet distribution. And for the genomic location marginal, we basically sample rather smooth functions from a Gaussian process with a stationary covariance function. And through this covariance function, we can really control how smooth those functions are and how rapidly we expect the signal to change. The smoother, the larger the neighborhood we can use to inform the origin of a given cut. And to ensure positivity, we apply the exponent here. After that, we assume that our final measurement, our data, is distributed according to a linear combination of these two target-specific probability density functions. And typically, you would try to infer certain weights for the two target-specific distributions. But the beauty here is that the Gaussian process itself isn't constrained through an integral, and the average value can basically pick the right weight for us. However, this also comes with a complication, because the Gaussian process sampled functions are not necessarily probability density functions and do not integrate to one, which allows them to just grow indefinitely when we try to maximize the probability of our measurement. So we really have to impose an additional constraint that makes sure that those functions actually are well behaved. Here we use a soft constraint for that because it makes it much easier for us to later on sample certain parameter distributions. And I'm happy to answer any questions about that. Unfortunately, Gaussian processes don't scale very well with a number of data points. And in our case, those data points are unique cut sites throughout the genome. However, a few things here come to our advantage. The first being that we only have discretely many cut sites at integer base pair locations, and that many cuts that we measure happen at the exact same base pair, which we hence only need to incorporate once into our deconvolution. The second thing that helps is that we use the Gaussian process only as a prior to sample smooth functions from, without conditioning them on any pre-existing data, and we only have to calculate the covariance matrix once and the respective Cholesky decomposition. So the complexity is not cubic but only quadratic for our application. And the last thing that really helps is that not all cuts do impact all other cuts. If two cuts are very distant from one another, there is basically their impact can basically be neglected in the deconvolution. So what we did to make use of this, we split the genome up into individual intervals. We leave out areas where there's basically no measurement, and we pick a maximum number of unique cut sites, which basically determines the maximum amount of work one has to do for a given interval. And then we group those intervals together such that every work chunk that consists out of a set of intervals has a representative set throughout the genome, and it's not biased because it only deconvolves a very limited area of the genome. To make sure that we can actually deconvolve even long stretches of very dense cuts, we would even subdivide those intervals, like in these examples. And we would make sure that those subdivisions overlap sufficiently such that the results all the way up to a given seam, here indicated by this black line, are not impacted by the fact that we ignore any cuts that are further out. So after deconvolution, we would basically remove part of the overlap on both sides, and the joint result of those two subdivisions can then be glued together without any weird jumps in our deconvolution signal. Now we can use this model for variational inference to estimate the latent distributions, the latent marginal distribution, and also the target-specific fragment length distributions. And one of the major results is, of course, the location marginal, which I want to look at here. So this is again just an example locus that I picked to illustrate a part of the results, which are these deconvolved tracks here for the polymerase and for the K27. And as a reference, I also show some single antibody data that is much closer to the ground truth to see how close we get. And of course, the 2-for-1 signal, the read coverages that we put in as deconvolved data. And these deconvolution tracks span the whole genome. And we can now do some peak calling, which is basically to identify regions of high intensity, which we can use as features for downstream analysis. We have a slightly different situation now than previously because we do not have read coverages anymore. Instead, we basically have cut probabilities for any location in the genome. And so we have to employ a different way to call those peaks. The way we do this is based on the amount of cuts that we expect to be within genes, and also based on the genome-wide integral of our results. So we can basically measure the share of cuts that we expect to be from one target or the other, and then select a cutoff for our probabilities such that the amount of cuts that ends up within the called peaks is actually the expected number, or the number that we expect prior to this peak calling. And you can do this for the two tracks independently, and then we end up with certain peaks here, and those peaks can of course also overlap. So to evaluate the quality of the called peaks, we can use the single antibody data again. Here I'm showing all the peaks that we called based on the deconvolution from the 2-for-1 signal, and that we say should be polymerase ones. And indeed, the polymerase signal from the single antibody experiment is a lot stronger here, and we seem to identify the great majority of peaks correctly. The same is true for the peaks that we claim to be K27, because the single antibody data again largely agrees with the result. So what we did here is we completely removed fragment length as the feature for the deconvolution. And the only thing that's left is the Gaussian process that models the location marginal. And the difference between the two targets is only in the covariance function. So for the polymerase-specific peaks, which aggregate in small peaks, we use a covariance function with a shorter range that can produce functions that change more quickly. And for the K27 signal, we just use a covariance function with longer ranges, so it should produce smoother functions. And the model would still come up with something reasonable, although there are many more false positives. It doesn't completely come apart. So apart from just the fragment length, the Gaussian process that just models the location marginal seems to play a very important role here. Similarly, we also wanted to illustrate the importance of the neighborhood information for a certain fragment. And of course, to do this, we would modify the covariance function of the Gaussian process and reduce the length scale drastically, such that the Gaussian process would basically not allow to incorporate any neighborhood information to make a call on an individual cut. So the location marginal becomes very noisy, and we identify way less peaks throughout the whole genome. So when it comes to the fragment length distribution marginal, it is a very important part for the deconvolution to work efficiently, but it's not really the result we are going for. It's still informative to look at them just to see how well or reliable the deconvolution worked. And as I said earlier, we don't really need to set a strong prior for the two fragment length distribution marginals. In this experiment, we still set a different prior for the polymerase specific reads, which have a higher first mode, so they tend to be shorter. And for the H3K27 specific reads, the first mode is lower, and the second mode, a little hidden here, has a higher frequency. However, when we run the deconvolution on this specific dataset, we can see that all the work chunks, which are run completely independently, produce a very similar posterior fragment length distribution that is very dissimilar to our mean prior. So the fragment length distribution is informed through the data, through the co-occurrence of certain fragment length throughout the genome, and is very well reproducible from different parts of the genome. Now I want to come back to our original deconvolution results and show you that we can in fact use them to make distinctions between biologically different cells. On the left side, we only use peaks that we attribute to the polymerase binding to the genome. We can see that the two cell types cluster nicely, and the same can be said using only peaks that are associated to the silencing HK27ME3 histone modification. In conclusion, we can say that single-cell CUT&Tag 2-for-1 can indeed produce measurements for both targets of interest. And we showed how we can use the fragment length information to reconstruct from those measurements which parts of the genome are more active and which are more silent. We also showed how important it is to use some neighborhood of any given cut in order to inform its likely target. And we think that single-cell CUT&Tag 2-for-1 can indeed help to illuminate the interplay between activity and silencing within the same single cell. And with that, I want to close my talk and thank you very much for your attention.